Research Center for the Archaeoscience of the Ancient Mediterranean, pronounced in Germany as MHAAM, and in the United States as MHAAM, or sometimes Mayhem. And under the aegis at Harvard of the Initiative for the Science of the Human Past, and with the support of the Departments of Human Evolutionary Biology and the Standing Committee on Archaeology. Our overall project aims to bring together the power of the advanced sciences and the questions and inspirations of the humanities and the social sciences to illuminate the human past and the microbial past, in which our present is, whether we wish to acknowledge it or not, so deeply embedded. In its initial research program, the new research center between Max Planck and Harvard is focusing on one hand on reconstructing ancient migrations in the Mediterranean, the peopling of the Mediterranean basin, from the genetics of the ancient DNA of those people, and on the other, in reconstructing and studying ancient pathogens and their evolution and their historical impact. In our inaugural meeting at last October, at which some of you were happy enough to be present, lucky enough to be present, we heard a lot about ancient genetics. We heard the first genetic evidence about cultural groups who have been archaeologically identified as Mycenaeans and Minoans. We heard about one of our favorites, Yersinia pestis, and more. Today we focus on ancient pathogens and on another ancient pathogen. Tonight is a special occasion in the life of our project, for this presentation offers the early fruits of a human collaboration between our Harvard scholars, our Max Planck scholars, and scientists and scholars from other institutions as well. It offers the very practical fruits of this unprecedented collaboration across the ocean and across the borders on our own continent. The devastating impact of the invasion of European life forms on the naive populations of the Americas are well known in a general way. And some of the terrible later outbreaks, for instance of smallpox in these regions, are historically well documented. But much remains completely unknown or controversial because historical accounts are incomplete, misleading, or ambiguous. The new archaeological science is now identifying ancient molecules of specific pathogens and assessing their impact. This is essential to reconstructing what really happened at various moments in the Columbian encounter and to weighing its historical consequences. These terrible events were tragic, beyond tragedy, for those who experienced them, and obviously of grave importance for historians and archaeologists. And I would say even for citizens, for they continue to affect in some way, direct or indirect, those of us who live in the societies that have emerged from this historical and biological crucible. But it is also of import for the modern day life sciences, and I hope we'll hear more about that as we go forward. Evolution is not something that started with the rise of microbiology. The pathogens that afflict us today continue constantly to evolve in the unending war between old drugs and new bugs. Many think that the bugs are beginning to get the upper hand. In that context, it is of more than historical interest to identify and understand the genomic details of the past pathogens. How did they get to be the way they were? And how do they differ from what they are today? What lessons does past evolution of pathogens hold for their future evolution and forms, and for our future medical responses? Tonight, we'll hear from my co-director of the German end of our magnificent new transatlantic collaboration, my treasured friend and colleague, Professor Johannes Krause, founding director of the Max Planck Institute for the Science of Human History at Jena, who needs little introduction to most people in this room because this is the fourth time in 14 months that I've had the honor and pleasure of opening a talk by him. To open the discussion, we will be privileged, after Johannes has made his presentation, to have from this side of the river a member of the steering committee of the Initiative for the Science of the Human Past, the Landon Clay Professor of Scientific Archaeology, Noreen Turos. From across the river, we are thrilled to have Dr. Ed Ryan, Director of Global Infectious Disease, Harvard School of Public, at MGH, Professor of Medicine. He's got too many titles here, HMS. Professor of Immunology and Infectious Diseases, Harvard School of Public Health, who works on enteric infections, cholera, typhoid fever, particularly in Bangladesh. We're no less thrilled to see so many distinguished colleagues from the medical school and from the School of Public Health, and we know that your presence here will nourish some wonderful questions and subsequent discussion, not just in this room, but in the hours and we hope the days and weeks and months that lie ahead. As always, we'll conclude with a short reception where our conversation can continue. But with no further ado, let's hear from the main act, Professor Johannes Krause, who will be talking to us tonight about death by contact, ancient pathogen genomes from epidemics in early Mexico. Please welcome Johannes.
Just 5 seconds for setup here. Is the microphone working? Can you hear me in the back? Okay, wonderful. So first of all, I would like to thank Mike for the invitation to speak again here. I'm kind of a constant visitor here, it seems, so I'm very excited to be here. This is work today that I'm presenting, which is actually not so much part of the Max Planck Harvard Research Center for the Archaeoscience of the Ancient Mediterranean. But it's actually a collaboration which we already started before we actually initiated that center, together with Noreen Touras, who will speak to you also after, which is a project where we wanted to study and find out more about the pathogens that were present in the 16th century in Mexico that caused some of those traumatic epidemics that diminished the Native American population in this part of the world, as many of you know. Before I talk about this results and this investigation, I would like to introduce you to our institute, because that is an institute which we just inaugurated also just a few years ago, in 2015 in fact, which is an interdisciplinary institute that nicely shows how different disciplines from the bioarchaeology are actually working together. It's an institute that has, I guess that doesn't work on this surface. So we have 3 different departments, archaeology, linguistics, and archaeogenetics that work together. They're an interdisciplinary approach to look at the science of the human history, similar to the Science of Human Past initiative here, which basically means in our case that we are using scientific approaches to classical questions that you might have from archaeology or history, which often involve the natural sciences, something like genetics, proteomics, isotopes, to address questions that can be, for example, mobility, so settlement history from the past, using genomics or isotope work, for example. And we're also looking at climate change and environmental change, so how do humans, for example, get affected by environmental change, and how humans, for example, also changed, of course, the environment over time, looking at sediment cores, for example, ice cores. We're also doing a lot of work on human modifications of the ecosystem. So we have a research group that is quite interested in how humans, for example, modified the rainforest over the last 10,000 years. For a lot of people, it's not quite clear that the rainforests have actually been affected by human activity over a very long time period and modified also by humans using, again, genomics as well as radiocarbon dating. We have Department of Linguistics and Cultural Evolution that has a strong focus on language diversification and the history of languages and how languages evolved and diverged from each other. So they're using actually tools that come from bioinformatics, from phylogenetics, and apply them to languages, so computational linguistics, by, for example, reconstructing then family trees of languages like the Indo-European languages, trying to find out when, for example, was the common ancestor of all Indo-European languages or Austronesian languages, and doing similar things also for cultural evolution. We have a strong focus also on the co-evolution of genes and culture. So how did basically culture influence genes, say practices, subsistence strategies such as agriculture, for example, then lactase persistence spreading within a population. And then that will be the focus today of my presentation, as we are also interested in the spread and diversification of human-associated microbes, which can be basically the microbiome, so microbes that live in you and on you, but also infectious diseases which are killing you at the end. So if we talk about infectious diseases, we have to actually go not too far back into human evolution. So the history of most infectious diseases is starting sometime, we think, in the early Neolithic. So about 8 to 10,000 years ago, when humans started to settle down. So humans basically changed their subsistence strategy from hunting and gathering to agriculture. With that came basically a kind of type of living in settlements, which caused them to settle down. And this kind of larger population sizes that came out of that then caused basically very good breeding conditions for pathogens to emerge within the human population. And what also happened is that humans started to domesticate animals, and it is thought today that from those domesticated animals, many pathogens actually then moved into the human population. That's a process which is called zoonosis, and there are many examples of that, such as measles, smallpox, flu, tuberculosis, plague, leprosy, or pertussis, where today we find the closest relatives of those human diseases or pathogens that are found in the human population in those domestic animals, for example. And then when the human populations grew in size, so large civilizations emerged, those pathogens found even better breeding conditions, and they caused massive outbreaks. So they caused epidemics on a larger scale, continental-wide scale, for example, that we then call pandemics. So large outbreaks which are then also found in historical records. Some of them I'm listing here, where, for example, the first and kind of earliest records we have are coming actually from ancient Greece, from the 5th century BC. That was the so-called Plague of Athens, which was a major outbreak that happened during the Peloponnese War. Actually killed the king of Athens at the time, Pericles. It's not really clear what pathogen caused this Plague of Athens. It's thought that it could have been measles based on the historical accounts, but we don't really know for sure. And then there's two large outbreaks of pandemics that happened in the Roman Empire, so the Antonine Plague as well as the Justinianic Plague. The Antonine Plague, some people think, has actually stopped the expansion of the Roman Empire, so again had a strong impact on human history. And the Justinianic Plague, which Mike is actually an expert on, in the 6th century, ended maybe the Roman Empire, or was one of the reasons that ended the Roman Empire. It certainly marks the transition from antiquity to the Dark Age, the medieval time. And that is one of the few then pandemics that we know from ancient history where we actually know what was the culprit. We actually know what pathogen caused it. Likely that was Yersinia pestis causing bubonic plague, because we've actually done genetic research on that quite actively over the last few years. And then there's two more that I would like to mention. One is the Black Death, which is probably the biggest pandemic that we know of in human history. It killed about half of the European population in the middle of the 14th century. Again, there, based on genetic research, but partially also historical research, we have now a good understanding what pathogen caused the Black Death. That was, again, Yersinia pestis, causing a bubonic plague pandemic. And then a pandemic which is actually not over yet. So some people say it's still ongoing, which is the White Plague that started in the 16th century, which is basically tuberculosis spreading within the human population, starting in Eurasia and then also spreading into other continents, which is caused by tuberculosis, or Mycobacterium tuberculosis. Now, except of tuberculosis, many of those pathogens that might be involved here, bubonic plague, we have leprosy, measles, smallpox, especially smallpox, which is actually eradicated, sound a bit like something from the past. And you might actually think that infectious diseases are more something from back in time in history and not so much more a topic of modern day medical research. And this is actually what also a lot of researchers thought in the mid of the 20th century. So in the mid of the 20th century, there were many medical researchers that were actually thinking based on vaccination programs, based on antibiotics that were developed, that by the end of the 20th century, we will not have infectious diseases anymore within the human population. Of course, today we know that this was not true. We actually know that things have actually gotten pretty bad and grim, because we actually see new pathogens emerging since the 1980s, which is partially due to the globalized world, partially also due to antibiotic resistance, that some of the infectious diseases are actually coming back. So we have newly emerging infectious diseases, many of them you of course know, HIV, Ebola, Hantavirus, and so forth. But we also have this antibiotic resistant bacteria, like multi-drug resistant tuberculosis, for example, which are becoming more and more a problem in the health sector. So infectious diseases, therefore, are still very much in the focus of a lot of medical research. When I started to work in this topic, I actually noticed, however, that even though it's a very hot and widely debated and important topic, we know surprisingly little about the evolution of pathogens. So we know little about evolutionary rates, so mutation rates, so how fast do pathogens change over time? So how fast do they accumulate mutations, for example? We also know surprisingly little about host-pathogen interactions. So how does an animal pathogen that was a pathogen in cows or pigs or dogs become a human pathogen? So what has to happen to this pathogen? And surprisingly also, we don't know so much about what pathogens caused epidemics and pandemics in the past, right? We don't often know what was actually the causative agent of a past pandemic. And the reason why we know so little about that is largely because if you want to study evolution, you usually have to look at fossils, right? So if I want to study the evolution of humans, I can look at Neanderthals or Homo erectus or Australopithecines. If I want to look at the evolution of elephants, I can look at mammoth or some other type of proboscideans. But if you want to study bacterial evolution, you don't really have fossils from the past. If you have a fossilized bacterium, it's just a round shape in a piece of stone or something like that. It's not really something that tells you something about the kind of larger biology of past bacteria. And this is when we then started this new field of research that we call ancient pathogen genomics. So we are basically creating molecular fossils from ancient pathogens by going back in time, taking skeletons from the past and extracting the DNA of pathogens from those ancient skeletons. And this is something then we do in our department for archaeogenetics, where we work with bones in clean rooms, where we dress up with protective clothes to avoid contaminating those ancient bones that we're analyzing with our own DNA. And we get this DNA out of those bones by just drilling in little holes that can be into bones, that can also be into teeth, depending what type of DNA that you're looking for. So if you're looking at the DNA from the individual itself, from whatever the human or the mammoth or the bear that you're interested in, you would just look at the bone. If you want to study the pathogens, we're actually not looking at bones, but we're looking at teeth, because teeth often still have dried blood inside, and in the dry blood you would find the blood-borne pathogens. If you want to study the oral microbiome, you might look into dental calculus, which is still stuck on the teeth. So it really depends what type of DNA you're interested in. That depends then which material you're sampling. And then from those basically little bone powder that you might drill out from those ancient bones, you extract the DNA, simple DNA extraction protocols, and then it gets turned into DNA which can be manipulated, sequenced, as you will later on see, also captured or enriched for the type of DNA that you're interested in. And then at the very end, that DNA gets sequenced in those wonderful modern sequencing machines, which, as many of you know, have kind of tremendously improved over the last decade or so. So the throughput of those machines has increased by 100 million fold in just about 12 years, right? Which allows now human genomes to be produced for several $100s, which 15 years ago was $2 billion for the first human genome. So it's really amazing how this throughput has increased, and it's very useful also for our ancient DNA, because the sequencing length that comes out of these machines is actually perfect for our DNA that comes from the ancient fossils, because the DNA after thousands of years is broken into small pieces, and those machines can only sequence short DNA molecules, but for us that's not a problem because our DNA is already short. So by then extracting the DNA from those ancient bones, we can then get those ancient pathogen genomes, and that then really gives some sort of molecular fossil record for ancient pathogens. So we can then really look how did certain pathogens, certain bacteria or viruses look 500 years ago, 5,000 years ago, 15,000 years ago. We can look at long-time mutation rates, so we can directly calculate the mutation rate of the pathogen because we have a radiocarbon date of this pathogen. So we know how it looked like 1,000 years ago. We know how it looks like today. We can measure the number of mutations that happened in 1,000 years, which is something that's very difficult to do if you just look at modern bacteria, for example. We can look at host-pathogen interaction through time, so we can see how did the pathogen change, whether certain mutations that had to happen, that it kind of adapted, for example, to new hosts, or that virulence factors actually emerged. So this is something that we've especially done a lot for Yersinia pestis, which was the main focus of our research over the last few years. And then, of course, we can give some very interesting information to the archaeologists, to the historians. We can learn what type of pathogens were actually present in the past that caused those large epidemics. So the type of question that, for example, Mike might have, what caused the Justinianic plague? We can actually then take skeletons from these large outbreaks, trying to find out what pathogen were present. Or Noreen that had that important question about what happened in the 16th century in Mexico, where we can then try to identify what pathogens are actually present in those ancient skeletons from Central America. And we've done that on a number of projects together with some of the group leaders in our department. So first we started with Yersinia pestis. As I mentioned before, this was really the main focus of our research over the last few years, where we were able to reconstruct it from the Black Death, so from the 14th century, as well as then from the 6th century and then later centuries. And we've even traced back the history of Yersinia pestis now into the Stone Age. So the oldest genomes we now have are more than 5,000 years old from all over Eurasia, and we can actually really reconstruct the genetic history of Yersinia pestis quite well now, even looking at when it emerges as potentially a human pathogen from some sort of pre-bubonic form to the bubonic form, which is very exciting. We also looked at ancient leprosy, medieval leprosy mostly, that we reconstructed from medieval skeletons from different sites all over Europe. For leprosy or Mycobacterium leprae was actually quite exciting now that we see that there was a very large genetic diversity of leprosy in the past in Europe. Actually, the same diversity that we find in the world today, which currently makes us think that at least Western Eurasia, maybe even Europe itself, could be where leprosy actually emerged in the first place. Which is a pretty crazy idea if you think about it, because today it's not found in Europe anymore, but in the past it was present in Europe and it was extremely diverse actually. There were many different strains, quite some genetic diversity there. But we are not only looking at human pathogens, we have been also looking at plant pathogens in the past. So we, for example, reconstructed Phytophthora infestans, which is causing potato blight, so it's actually a plant pathogen. And this plant pathogen caused one of the biggest migrations in human history from the last 200 years. There was the Irish potato famine that caused a lot of Irish people to actually leave Ireland. That happened in the mid-19th century, and we were actually able to use herbarium collections, so leaves that were in herbaria, to reconstruct from those leaves from the mid-19th century this oomycete, so fungus-like organism, and then compare that to modern potato blight, so modern Phytophthora infestans, to see how it changed over the last 150 years. What we actually found was that the one that was causing the potato blight in the 19th century is actually extinct. It doesn't exist anymore. It was actually replaced by another one because potato actually got resistant against the one from the 19th century at the end of the 19th century, and then a new one was basically introduced at the beginning of the 20th century, which is now present in potatoes, again actually causing a lot of potato loss in the world. But this one from the 19th century is actually extinct because it was basically not as well adapted. We've been also looking at Helicobacter pylori, so the bacteria that can survive in your stomach, that can cause ulcer, for example, or stomach cancer. And we have been able to reconstruct them. You might wonder, how do you get ancient stomachs? There is actually one really nice preserved ancient stomach that comes from this gentleman here, which is the Iceman. So the Iceman that was stuck in the ice in the glacier of the Alps for about 5,500 years. Was discovered in the 1990s, early 1990s, and we had actually access to the dried stomach of, or freeze-dried stomach of this mummy, and we could indeed reconstruct Helicobacter pylori from this ancient Iceman. We've also done some work on Mycobacterium tuberculosis. This is a project I was actually thinking maybe to kind of cover today in the presentation, but I think it would have been too much. But this is also a New World pathogen that we have here reconstructed, but this is not post-contact, this is actually pre-contact. And that was very exciting because for a lot of time, people did not think that tuberculosis was present in the Americas before contact. And that is because if you today look into North and South America, the tuberculosis you find in native populations as well as kind of populations from today is they have usually tuberculosis of the European type. So tuberculosis or Mycobacterium tuberculosis has basically different types. You find different parts of the world. There's some sort of phylogeographic pattern. There's a European version, there's an Asian version, there's a lot of different African versions. The highest genetic diversity is in Africa. But what you find in the New World is all basically the European type. And because we know that the Americas have not been settled, pre-contact at least, from Europe, but actually from Asia, it was kind of strange that you find just the European type, and therefore people assumed that tuberculosis came with the Spanish in the 16th century to the New World. But our colleagues had actually identified that there are human remains from pre-contact North and South America that actually show tuberculosis lesions in the skeleton. So they have very clear pathology that indicates that tuberculosis was present in the New World before contact. And our question then was, what type of tuberculosis was that? And now, to our surprise, when we reconstructed it, the type of tuberculosis that was present in the New World before contact was actually not a human tuberculosis. It was actually not tuberculosis bacteria you find in humans today. It belongs to the Mycobacterium tuberculosis complex, but it was actually a version that today we only find in pinnipedes, so seals and sea lions. That was a big surprise to us, but it might actually explain how it came to the New World, because what we also found was that tuberculosis itself as a complex, so all tuberculosis bacteria that are found in the world today, if you reconstruct their most recent common ancestor, and because we have those ancient genomes, we can now have a good mutation rate, we actually know how old tuberculosis roughly is. It's only about 6,000 years old. So it's a very recently kind of spread infectious disease, probably also in the Holocene in the last 6,000 to 10,000 years. And that was always hard to then imagine how it got from the old world into the new world in the last 5,000 years. But now we might have a good explanation because those guys, of course, can swim, right? So it was basically swimming from Africa, likely into the Americas, and then disseminated in the human population. And then the last one, and that's kind of should be the focus of the presentation today, is the one that we just recently reconstructed, and that is Salmonella enterica from the New World. So the New World history, of course, as you all know, we are the New World. It's a very exciting history, but has also seen, especially in the first decades, quite some devastation in the Native American population. Because when the Europeans came to the New World, they brought diseases to the New World that those people that lived in the New World had not been exposed to before, at least not for a very long time. And we think today that those diseases might have then also facilitated the European conquest because they just killed, they were so devastating within the Native American population that this population plummeted, and then basically the Europeans had the chance to basically then settle and basically take over, if you want, especially North America and Central America. So the estimates go that about 95% of the Native American population were killed by infectious diseases in this first 100 years after contact. Some of them we know based on the kind of description from that time period. For example, like smallpox, that was in the 1520s, a smallpox epidemic in Mexico, which based on the description was very likely smallpox. Measles, mumps, influenza have also been indicated, maybe in later centuries, were responsible for some of this large epidemics that happened. But there's still much debate about what other pathogens might have been present in the Native American population and which other pathogens were brought from the Old World into the New World. At least for Mexico or Central America, we have three major epidemics where we don't really know what pathogen caused them. And those are called the Coccolizli. Coccolizli itself is actually a term from Nahuatl, that means just pestilence. So it doesn't really tell you what type of pathogen it is. It just tells you that there was some outbreak, some sort of plague outbreak, some pestilence-like outbreak, which is not really clear what it was. If we look at the numbers, you can actually see how strong those 3 epidemics influenced the Native American population. So we started off with something like more than 20 million people that lived in Mexico at the beginning of the 16th century, and we ended up by the end of the 16th century with about 2 million or a bit less than 2 million. So really like more than 90% of the population, based on historical accounts, were basically killed by those large pandemics. And the biggest of them was the Great Kokolitzli that happened around 1545, lasted maybe about 5 to 7 years. And then there was the second Kokolitzli that happened in 1576, and then the third one, which was 1590 at the end of the 16th century. And there's been much debated about what type of pathogen caused them. There's some very interesting accounts, if you're interested, from Alexander von Humboldt, for example, who was debating in the kind of early 19th century what type of disease might have been responsible for it. He concluded, based on the historical accounts, that it was some sort of typhoid-like disease, which is quite interesting because, as you will see, that came pretty close. At the time, typhus and typhoid fever-like diseases were not yet differentiated, so it was actually interesting that he came to this conclusion. So what do we know about the Great Kokolitzli? It's a disease, a pandemic, or epidemic, large epidemic, that not just affected Mexico, but larger parts of Mesoamerica. Some people even think that it also went into South America, that affected parts of Peru, for example, and Colombia. From the historical records from later time periods, you can reconstruct some of the symptoms that people had. So there was some sort of blood-borne fever, there was bleeding from facial orifices, there was rash, there was diarrhea. It not just infected the indigenous population, but it also infected the European population. So it was not something that was just spreading within the native population, but also the Europeans were infected. It's not really clear from where it came from. So some people think it could be European, so a disease that was introduced by the Spanish, but there's also people that think it could have been some sort of local origin. So some people have claimed that it could have been some sort of virus that emerged from within the Native American population, or within the Americas itself, and then spread within the population, and it was actually nothing that was introduced from the outside. Why do we know so little about it? That is largely because we have very little historical records from that time period. If we actually look at the historical records we have directly from the Great Kokolitzli, then this is all we have. That's basically the historical record. That is this one drawing, which is from the Codex de la Cruz, which is just a person that is dying, probably with some sort of liquid coming out of its mouth. Right? That's all information we have. And if this liquid is now blood, if this liquid is some other body fluid, is not really clear. Only in later periods, so 20, 30, 40 years after the Great Kokolitzli, we have some more information, some more codexes, which are mostly drawings and some writings about what happened in the past, in the basically 20, 30 years before, during this pandemic. Then you see also colorful images where you then have, for example, blood or some sort of bloody liquid coming out of people's mouth, which again looks maybe some sort of blood-borne pathogen, some sort of hemorrhagic fever, for example. And we have then also from later on accounts this type of drawings where basically people then report that there was massive amounts of people dying during this time in 1545, 1544, 45. And based on this limited historical records, people have then suggested different types of pathogens that might have been responsible for the Great Kokolitzli, so that it was some sort of hemorrhagic fever based on the blood or the kind of bleeding. People then still said it could have been some other disease like measles, smallpox, or typhoid-like diseases, for example, or even pneumonic plague, some people have suggested as well. So there's been all kind of hypotheses what Kokolitzli actually was. And this is then basically where we started off, together with Noreen, who had actually been working with a colleague from Mexico, so Nelly Robles Garcia, who had been excavating a site that is called Tepos Kulula Yucunda, which is a site that is a former Mixtec site, which is in Oaxaca in Mexico. And they had actually excavated the site. And when they excavated the site, they actually found a large mass burial in the center of this former Native American town. So this was a cemetery which is actually here present on the Grand Plaza, which was a cemetery where more than 800 individuals were buried. And they were buried in a very interesting way. People had basically dug holes, long rows, basically entombed those people, but hadn't really closed those holes. So this marketplace was basically full of big holes when the people left, so they didn't even fill it up, so it was never used as a marketplace again. Actually, the town was even given up, so the site was abandoned just after this epidemic and was not used again. They actually moved the entire town to another place. So it was such a big event that happened here during this Kokolitzli. Probably so many people died that they actually gave up that town and moved somewhere else. Maybe they also thought there's some sort of bad spirits, not really clear, but people abandoned the site. And we then analyzed human skeletons from two different parts of the site. So first of all, from this mass burial, from actually the only mass burial that we have from the Great Kokolitzli, from this then time period, 1545 to 1550, as well as human remains from the so-called churchyard, which is actually a pre-contact cemetery, which was also present in this village. So we got skeletons from the pre-contact site as well as from the post-contact site to see what type of pathogens we might actually find in those individuals. Then my student Ash, who was actually working on the analysis, she then looked at those ancient samples. So she used teeth again, because as I mentioned before, teeth have the dried blood, where we can then extract DNA, and that DNA can then come, of course, from different types of origins. And in this case, of course, unlike, for example, for the Black Death, where we might have had some idea what pathogen might be present in those individuals, we had no idea what pathogen we could actually look for. And therefore, we choose an approach which we call a shotgun approach, where we extracted DNA from those teeth and then looked for what organism, what bacteria those DNA fragments that we can extract from the ancient teeth can actually come from. And this in principle should be straightforward, but it's not completely easy, because you're usually producing with this next generation sequencing quite a large number of DNA molecules, like millions, that we produce from each tooth. And then to kind of identify what organism those DNA fragments are coming from is quite challenging, because it's just computational, not completely straightforward. And therefore we used a tool that we had developed in our lab, which is called MOLD, which is a really fast way of basically identifying what organisms those DNA fragments come from. And this is a tool which is called MHAAM alignment tool. So MHAAM is a metagenomic software that the people that know metagenomics might have used before. And it's actually quite a fast, rapid tool for aligning DNA fragments to a reference database. So you can use thousands of genomes. So in this case, we used about 6,000 bacterial genomes. We have now also used all of GenBank, which has actually also mammalian genomes as well as viral genomes. And you can, in parallel, basically screen all the DNA fragments against this large database. And it's quite fast. As you can see here, it's about 200 times faster than BLAST. BLAST is, for the people that don't know, BLAST is kind of the gold standard alignment kind of taxonomy identifier that people have been using in the past. So it's a bioinformatic tool that's, for example, available in the web, where people, if they have a DNA sequence, blast that DNA sequence to see what organism that's coming from. So it's something that's been around for more than 20 years now, but this is quite a bit faster and allows you rapidly to screen a lot of DNA sequences. So then the idea is basically we take the DNA from those ancient skeletons and then just shotgun sequence that and look with this algorithm how it looks like. So this is just the principle of this algorithm. I don't want to go too much into detail here, but what we basically have is our raw sequencing reads. We compare that to the MOLD database, and you can choose whatever database you want to have. In our case, we looked at all bacterial genomes because we were interested to see what type of pathogens might be present in this data set. And you then get some sort of output files which can be in different formats. that you can then, for example, feed into MHAAM. So MHAAM, again, is this metagenomic alignment tool where you can basically see what type of DNA comes from what type of organisms inside your sample. Or you can also have some output files, SAM format, which is a standard output file that people use for alignment. So it really produces a DNA alignment. So for people that do bioinformatics, it's like BWA or some other alignment programs. It really produces alignments that you can actually look at, which is quite different to Kraken or some other metagenomic tools that people are using currently. And it's very fast, quite efficient, because what it actually does is it kind of produce some sort of hash tables which it loads into the working memory of the computer. So you need actually a computer which has about 2 terabyte of work memory, which is not the normal computer that most people have, but still most bioinformatics centers have such a computer available. And it can process about 1 billion reads in less than 24 hours, which is extremely fast. If we would have done our work with a different kind of algorithm, not with that type of approach, it would have taken months, but in this way it only took hours to screen basically this larger data set from this Kogolitzli site. The output that you get looks like this, which I guess hard to see in the back. It's even hard to see in the front. So this is a gigantic phylogenetic tree. So basically each node here is different bacteria that you can find. That's just for some random sample that we had from a tuberculosis screen. And what you see is actually thousands of bacterial kind of clusters that are supposed to be present in this sample from this bone that this DNA has been extracted from. You can also zoom in, then it looks like that. This is just for mycobacteria. And then you see that you have supposed to have all those different types of mycobacteria present here. So not just Mycobacterium kind of in general, but bovis, Candidii, tuberculosis, leprae. So everything is supposed to be in this one sample. And this is true for almost every metagenomic sample. So if I take dirt from the table here or from the backyard, you always find all kind of DNA from bacteria that is similar to bacteria in larger databases. And that is largely because most bacteria in the world have not been sequenced yet. Right? There's probably billions of different taxa or OTUs, how they're called, from bacteria. But there's only about 6,000 that were in this database. I think there's about 12,000 that are sequenced so far. So it's a very small number of bacteria that are actually known. Most of them are human pathogens because that's where our main interest is in, but we are not sequencing a lot of environmental bacteria, for example. So if you take an environmental sample and you compare that to a database, you will find all kind of human pathogens, but it's actually not the true human pathogens that are in the sample. This is actually largely false positive. So it's just some similar bacteria that have 90% sequence identity, 85% sequence identity, but are not really truly that type of pathogen. So it doesn't mean if you find this here that it's there. So you have to have some other criteria to make sure that this is indeed the pathogen that you might be looking for, that it's a true positive hit and it's not a false positive hit. And we then established a pipeline, which is actually not published yet, which we call RMA extractor, which takes the output file from this mold and then does a number of steps to make sure that the DNA that you have found with some sort of taxon ID is indeed from a pathogen that you might know of, and it's not some sort of close relative. And one way to look at that is edit distance. Edit distance just means the number of basically mutations away from the DNA sequence you have. So if you take your DNA sequence that you have, you compare to the reference sequence, how many basically substitutions, how many mutations, how many steps away is the DNA that you're studying. And if you have 0, it means it's identical. If you have 1, it means it has 1 substitution, 2, 3, 4, and so forth. And if you have something that's very similar, it should have an edit distance plot that kind of looks like this. If you have something that is more different, has a lot of substitution, you have something that more looks like that, which then tells you it's a false positive. So that's one way to look at it. You can also look at the read distribution. So what you want to have is if you have a reference sequence of a genome, you want to have that your fragments actually fall all over the genome and not just in one particular part of the genome that is highly conserved. There are elements inside bacteria that are conserved between many different bacteria. And if all your reads end up in this one place, you actually know it's not that pathogen that you have there, but just a close relative that happens to be similar in this part of the genome. You can also look at DNA damage. I will come to that a bit later. And you can then also take the DNA, which has damage. So this is DNA which has changed over time because it's old, it's ancient. And you can then look at this ancient DNA and then look at the added distance of this ancient DNA, the DNA which is actually old, and it should also have this type of pattern here and not kind of some sort of normal distribution. And if you take these different filtering steps, you can then screen also larger data sets. We've done that in this case for about 2,800 human samples, so from different time periods. So this is DNA now, not just from the pre-Columbian or post-Columbian, but this is actually from all over the world. Even Neanderthals are in here, Paleolithic, Mesolithic, all kind of samples that we have been screening to just see what type of pathogens we have in here. If we apply this MOLD tool, if we apply those filtering steps, we found in this case about 132. Depending on which filter, actually many drop out, so no filters, you have 320,000 pathogens, which is probably mostly false positives, but then based on which filtering step you see, actually this number goes quite a bit down. And then to get an overview of what is in there, we have this kind of plots, heat maps, where you have the pathogens here, and then this is all the samples. This is just kind of part of those samples that come from the late Neolithic Bronze Age, so they're about 5,000 to 4,000 years old. And basically then, those are the 4 different filters. So the kind of warmest color here, red, means it's a good, positive, likely positive hit. And if you have something that is blue, then maybe it's a candidate, but you should evaluate that more, maybe mapping to that genome, for example, or specifically zooming in. And you can actually see that we have quite a number of, it seems to be ancient bacteria, which are good candidates, good positive hits present in those, for example, 5,000-year-old human remains. Most of those are not really pathogens per se, but they are actually commensal pathogens, so bacteria that live, for example, in the oral cavity, because most of those samples are teeth. And then you often have something like Treponema denticola or Streptococcus mutans, which can be pathogens, but they also live in your mouth. So those are, for example, like this guy here, which you can actually see many of them, as well as other guys. But you also see some of the pathogens that we're interested in. For example, this row here is Yersinia pestis, and there's actually quite a number of samples here from the Bronze Age, from about 5,000 years ago, that have positive hits for Yersinia pestis. So they give us some indication Yersinia pestis was present in those samples. And that is then actually something we followed up in this case, and we were indeed able to reconstruct Yersinia pestis from those Bronze Age individuals. But coming back to TPOS, our site from Mexico, from ancient Mexico, we did the same approach, the MALT approach, for the TPOS site. We had about 1 million DNA sequences that we compared with this large database. And then again, you get this massive tree, which is actually quite huge. At the time when we had that, we didn't have this nice filtering tool that I just presented, but actually Ash, the student, had to go manually through that list, kind of look at every node, and see if there's one node that is bigger than the other nodes, which might indicate there's more DNA of that particular pathogen, which was a lot of work. That's why we invented this other kind of new tool and approach, because this kind of manual approach takes a lot of time. And what she found, what basically stuck out, is that there was a large cluster of reads that were mapping to Salmonella enterica subspecies Enterica seroor paratyphi C. And that was quite interesting, because it's a very specific type of Salmonella, which was seemed to have been present here. And it was quite a number of reads. It's just 600, 700, it's not large numbers, but it's still big enough that you might wonder if that could be the pathogen that was responsible for the death of that person. We actually found 3 samples that had several hundred reads, and then about 7 that had lower amounts of reads. So if you then look at the summary for all the samples that she screened, you can see that in the samples in the post-contact Grand Plaza, there were about 10 samples that had actually pretty high fractions compared to the other bacteria that we find in those ancient humans that came from Salmonella enterica paratyphi C, based on this analysis. If you look at the pre-contact site, you can actually see that none of them was positive. So there was no indication of Salmonella enterica present in the skeletons from the pre-contact site. We also had one sample here that is actually a sample which comes from sediment. So it was sediment DNA. We just wanted to make sure it's nothing that comes from the environment. So that also came from the Grand Plaza. But in the sediment, we actually didn't find anything that looked like Salmonella enterica. So it was a good indication that Salmonella enterica was present there. And we wanted to then follow this up. One problem you have, if you then have an indication of that DNA that might be present, you want to make sure that the DNA from this ancient pathogen that you're interested in is indeed ancient, that it's indeed DNA that comes from the past and nothing that could have contaminated the site. Maybe there was some sort of people were fertilizing with, for example, feces of pigs, which is quite common, that often carry Salmonella. So we really wanted to make sure that this DNA that we have here is not from the present, but it's actually from the past. And to do that, we look at something we call DNA damage. I mentioned it before. It's basically when the DNA gets old, it breaks into small pieces. And it's not only breaking into small pieces, it also accumulates chemical damage. So the basic chemical structure of the DNA changes. There's something that we call a miscoding lesion, where, for example, one base of the DNA gets chemically so modified that during the DNA sequencing or the amplification, we actually read it as a different base. So one example is cytosine, so the C in the DNA, which can get deaminated. And when it gets deaminated, it turns into uracil. And uracil is read as thymine. So a C actually becomes a T in the DNA sequence. And that is something that happens especially in the ends of the DNA, because the ends of the DNA, it's often single-stranded, so the DNA is broken, so you don't have the double strand, but you have the single strand, and then you accumulate that damage, especially in the single strand, because it's not protected like the double-strand DNA. And if you look at long DNA fragments, so this was, for example, Neanderthal DNA we analyzed some years ago, you can see that the damage is especially strong at the beginning of the DNA fragment. So this is the 5' end of the DNA. So we see a lot of C to T's. So the red line here is C's that have become T's. And then it goes down. And actually, on the other side of the strand, we see G's that have become A's. When we first thought that, we were not quite sure why have we C to T at one side of the DNA and G to A at the other side of the DNA. But that has something to do with how we sequence the DNA. We actually have a step where we fill in the DNA. We're actually filling in this part of the DNA. And during this fill-in step, we're basically putting the reverse complement base into this part of the DNA sequence. So if you have a C to T on this side, you get a G to A on the other side. So what we actually see is C to T on both sides, but here we have the reverse complement strand of the DNA. And that is something that we see in ancient DNA. Like this is Neanderthal, this is 40,000 years old. But we also see that in DNA, which is hundreds or thousands of years old. But where we do not see that is in modern DNA. So for example, this Neanderthal was contaminated with maybe my DNA or anthropologist or somebody else, and the modern DNA, the contamination doesn't have that. So it's basically completely flat. We also looked at that through time. So this is time here on the x-axis, and this is the amount of DNA damage on the y-axis. We can actually see a very nice correlation. So the older the DNA gets, the more damage it accumulates. So if we have, for example, DNA that has 20% damage, you can be pretty sure that it's older than 100 years. So it cannot be contamination. And this is something that we have been using over the last 10 years or so to really then authenticate if we have DNA from the past, whether that DNA has this damage pattern. If it does not have that damage pattern, we have to assume it's modern contamination. Because all ancient DNA we have analyzed so far has this damage, usually more than 10%, many cases more than 20%. And if you look at the DNA then from the 10 individuals that were positive for paratyphi C, we can actually look at the paratyphi C, and you can actually see those nice patterns here. We sometimes call this smiley plot because they make us very happy and they kind of look like a smiley. And you can actually see that all samples have this smiley. They have this damage pattern, which is typical of ancient DNA. So we can be rather sure that this Salmonella DNA that we have present here is coming from the past. So then the next step we wanted to do is not just see what type of DNA is present, but actually trying to reconstruct the entire genome of that ancient Salmonella. That's something that we have been pioneering in this first paper where we looked at the Black Death genome. And that's where we use probes which are bound to little pieces of glass, so capture arrays, to fish out the DNA that we're interested in. So basically what we have is a DNA capture array, little glass slide, with DNA that is synthesized on the surface of that piece of glass. That's something that companies can do for you. You can actually design those probes yourself. They're about 60 base pairs long, and you can have millions of them on the surface of this piece of glass. In this case, we used modern Salmonella enterica diversity. So you can actually see the probe design here. For modern Salmonella enterica, so 24 different genomes from the whole diversity of Salmonella, so not just enterica, but even close relatives, and put them all on this piece of glass to then incubate that DNA from the ancient skeletons with this piece of glass. The complementary fragments will bind to the DNA on the surface of this piece of glass, and you can wash away the background and then sequence the DNA in the next generation sequencing machine. And that actually works quite well. So if you look at the amount of Salmonella DNA that we had before enrichment was very, very low, just a few hundred sequences we had per million DNA sequence that we had sequenced, but then it was about 30%, so several hundred-fold enrichment. This is something that we are doing quite routinely on all kind of pathogens with this kind of capture. It also has the wonderful property that capture, unlike, for example, PCR, allows for sequence mismatches. So with PCR, you often have the problem that if there is a basically substitution in the priming site, you don't get a product. But with hybridization, even if you have 10% divergence, so even if the DNA sequence you would like to fish out has 10% differences for the DNA that you use as a probe, it would still bind and you could still fish it out, which is actually quite nice. And doing that, we got from 5 of the 10 samples genomes with pretty high coverage, so between 4.5 to 100-fold coverage, which means we have seen 100-fold coverage, for example, we have seen every position of the ancient Salmonella genome about 100 times. So that's actually pretty good quality, so the genomes are pretty much complete, and we have a very high confidence that the DNA sequence we have from them is actually the true DNA sequence, not affected by damage, for example, by the old age. So it's basically as good quality as modern Salmonella DNA that you would get from next generation sequencing approaches. What you can then do is family tree. You can actually see how those ancient Salmonella genomes are related to modern Salmonella genomes. And this is now all kind of Salmonella genomes here. So this is paratyphi, for example, then kind of outgroups like this Arizona sarovar here, typhimurium here, so something that you find in mice, for example. So all kind of different types of Salmonella, and you can actually see up here on the very top, this is where our TPOS samples fall in the family tree of Salmonella. They're falling very closely to paratyphi C. There's just a few hundred substitutions and mismatches or differences. And that is, on the other hand, then very close to cholera sus, which is actually a type of Salmonella you find in pigs, which can affect humans. It's actually pretty severe if humans catch it as a disease, but it's usually found within pig populations, and also caused some people to then propose that actually pigs gave this type of Salmonella, this type of paratyphi C Salmonella into the human population. So there was some sort of zoonotic transmission from pigs into humans that happened maybe a few hundred or a few thousand years ago. I don't have much time to present some new data that we now have from other ancient Salmonella. They actually suggest that it was not given from pigs to humans, but actually humans gave it to pigs. But something maybe we have time to talk about afterwards. So similar to tuberculosis, where actually we gave it to cows and not cows gave it to us, like a lot of people have thought. So this paratyphi C that we have here is actually just one out of 4 Salmonella enterica serovars that cause this type of enteric fever, so which is called paratyphoid fever or typhoid fever. So there's more than 2,600 serovars, so different types, different strains of Salmonella, but most of them are not causing some sort of enteric fever. They can cause some gastric disease, but they're not entering basically the bloodstream. They're not basically causing some sort of septic state, so they're staying outside or kind of inside the gut, but they're not entering the human body. It's quite rare today, paratyphi C. We don't find it at all in Europe, except of tourists that actually bring it when they have been in Asia, but especially in Southeast Asia, in Africa, it's actually still common today. But it's just a few thousand cases per year, so it's actually not as common as it might have been in the past. I mentioned it before, we have been looking for it now in our large data set. I showed you we screened more than 2,000 human skeletons from the past, and we have actually found it going back to 8,000 years in the past. And it's actually, besides Yersinia pestis, one of the most common causes of death that we can reconstruct in skeletons from the past. So it seems to have been very common in the past, not so much more in the present. It's transmitted fecal-oral. Maybe hygienic conditions have improved over the last few thousand years. I would hope so, at least. And that's probably why we don't find it so common today anymore, but it seems to have been quite common in the past. So it causes some sort of fever, body rash, as well as diarrhea in the mild cases, but it can also cause death if untreated with antibiotics. Today it doesn't cause so many casualties anymore because mostly people get treated with antibiotics, but it can still be quite severe. In untreated cases has a pretty high mortality. And there's also quite a number of people that are asymptomatic carriers, so basically kind of the typhoid Mary kind of case. I guess people have heard about it, like a person that was carrying bacteria over basically her entire life. In the beginning of the 20th century, she was an asymptomatic carrier, so she had those bacteria, but transmitted them without getting sick herself. And that's something that happens also quite in a few cases where people have this particular strain of Salmonella enterica. So concluding remarks, then what can we say? We've seen that about 10 of the 24 individuals that we looked at from this epidemic cemetery carried Salmonella enterica. The others also had reads to map to a Salmonella enterica, but not in large numbers. So it could be that all of them died of it, but it seems that at least half of them died of Salmonella enterica. So it's a very good, therefore, candidate agent for the great Coccolizli epidemic. I say here candidate because we cannot exclude that there were other pathogens present during that time. The study has been widely discussed and widely criticized also. It's always good to get critique, but people have basically said, why do you say now it was Salmonella? Couldn't it be still some hemorrhagic fever or something else? Of course it could be. We can only say that for this one epidemic outbreak in this town of Typos, we have the presence of Salmonella enterica. It could be that there were other strains of other pathogens that were present in the larger region of Mexico during the time, or that it was even a virus that caused a kind of hemorrhagic fever, such as a filovirus, like Ebola-like, that was present, what some people have argued based on the historical records. So we cannot really rule out that the pathogen actually existed before in the New World, so that it was not brought in by Europeans, but that there was some sort of local origin of it. But there's one very interesting study, which is actually not published yet, but it's available on the bioRxiv, which looked at Vikings and actually identified Salmonella enterica in 12th century Norway. So they have a strain of Salmonella enterica from 12th century Norway. So this is work by Mark Achtmann and colleagues from Warwick University. And interestingly, if we compare this Salmonella enterica from the Vikings, that fortunately is already available, and we compared it to our Salmonella enterica from Typos in Mexico, is actually very closely related. So they're falling kind of very closely together, suggesting that a very close relative of the Salmonella has present in the 12th century in Norway. And then, of course, everybody knows that Vikings have made all its way into America in the maybe 11th century or something like that. So they could have introduced it before, but we think that's an unlikely scenario because there has been only one settlement in Newfoundland. But likely it was present in all of Europe during this time. We've also found it in Southern Europe, actually, in the past. And the Spanish likely then brought it over, because it's very unlikely that the strains that we have in the New World in Typos are removed 20,000 years from this Viking strain, because they're too closely related. So basically we have it in Europe pre-contact, so likely was brought in by the Spanish in the 16th century. So there was, of course, a lot of trade, a lot of people arriving. There was also a lot of livestock that was brought by the Spanish into the New World, like pigs, for example. I mean, as you know, most native population didn't really have a lot of domestic animals, except of turkeys and llamas and guinea pigs. There wasn't a lot of domesticated animals in the New World, but then they introduced cows, they introduced horses, they introduced pigs, and it could even be that because of this introduction that the pathogen actually came with these animals into the New World, and probably also changed the whole way how people interacted with animals and among each other. What I've also shown you is this wonderful new tool MALD that, at least for the people that do metagenomics work, is actually quite a nice tool. It's extremely rapid, it's quite sensitive, so you can really detect small amounts of DNA that match to a certain reference genome, and it really allows now to find all kind of candidate pathogens from ancient skeletons that we or others might be interested in. What I also want to mention is, if you look at where do we have genomic DNA from ancient skeletons in the world right now, you can see that we have a very strong bias on Europe, because that's where most of the labs are actually based, and also where most of the samples are coming from that people have been analyzing. But you can actually see there's large parts of the world where we don't have a lot of ancient DNA that we can look at for ancient pathogens. So there's a lot of work we have to do. So there's a lot of basically future investigations for pathogens that might be present in other parts of the world. You can also see for Mesoamerica, North and South America, that's actually quite a small number of samples. I mean, it's still quite amazing to see that it's about 3,500 or so human samples that have been analyzed over the last few years. Given that when I did my PhD about 10 years ago with Svante Pääbo, we worked for 5 years on one genome. That was the Neanderthal genome, which was pretty low quality, and it was the first archaic human genome that was produced. It took us 5 years, cost 10 million euros, was a big project, and now we have actually thousands from all over the world. So we have made great progress, but still there's a lot of work to do, and maybe we can even do some of that work with some of you together. So by that, I would like to thank, of course, all the people that did the work and that just talked about it, like myself. The graduate students, the postdocs, Alexander Herbig and Kirsty Boss, which are group leaders now in our department. Noreen, of course, as well as Tina, who's a professor in our department, and Nelly, who provided us with the samples, the rest of the team, rest of our department, and all the funding sources. And thank you for your attention.
Thank you very much, Johannes. Let's begin with the first comment from my friend and colleague, Noreen Chouros.
I just have really 2 points to make, one of which Johannes has made beautifully. I just want to stress. This is really the first project where the pathogen was completely unknown. There was no morphological indication. There was only guesses based on historical records, which turned out not to be very helpful. This changes everything, and frankly, this is what I have been waiting for. The search for ancient pathogens that are associated with morphological change or with a good historic record are wonderful, but this is an order of magnitude different, and this is what we need to solve the issue of what killed indigenous peoples at European contact. Certainly, Salmonella enterico is not the only disease, but we have been completely hamstrung in trying to identify this and have, as Johannes said, only the records written 50 to 150 years after the fact in order to please the king that are of little value today. The second point I'd like to make is if you're going to get into this work, try to find a nice location. This is Tepozcalula. It's in the highlands of Mexico, which not only do you want a nice location, now that you've heard Johannes, you realize that this doesn't take a village. It takes several cities across many countries to pull off. And so when you're starting, it's probably good to start with a really good excavation of well-preserved materials. And as Johannes has pointed out, try to find a really excellent archaeologist, which we did in the form of Nelly Robles Garcia. So Tepos was excavated by Nelly and a very large team of people. And this is one of the graves from Tepos, and I want you to notice two things. One, this was carved into a flooring, a plaster flooring, which probably percolated down through a few hundred years and buffered the pH of the skeletons, and so they were magnificently preserved. The other thing you'll notice is there are 3 people in this burial. This was true at many of the burials in Tepos. People were dying so fast that they couldn't give them individual burials. This is sad, but when you find this, it's a very good indication of an infectious epidemic. The indigenous people were brought to the top of this mountain by the Dominicans to help build a church. And the epidemic ensued, and the deaths were so fast that, as Johannes said, the top of the mountain was abandoned. And today, and the last comment I'd like to make, is that the other reason Tepos and Tepozcalula is really important is that it's the gift that's going to keep on giving. This collection is available in large amounts in my laboratory. It was taken at excavation by gloved hands and put immediately in a freezer and taken to the United States. The rest of the collection is in this building, which when the Dominicans left the mountain, they came down to the village and built this church in what is now the village of Tepozcalula. And what you're looking at is at the outside chapel, which has been restored by the Mexican government. It's an outdoor chapel because the indigenous people were, although they were welcomed into the Catholic faith by the Dominicans, were not allowed inside the building. And so they had to stand on the lawn in order to receive communion. And so the indigenous people that helped build this were never actually allowed to go inside. However, the remains of their ancestors are now stored inside the building, something I take great pleasure in telling you. We continue to work on this site, and I would just stress how important it is to be able to go back to material and keep reanalyzing it, because there are people in this room who will have new ideas, and if we can build on collections and information, this is going to be transformative. I think a lot of credit to Johannes and his group, and to Alexander Herbig, and for the development of malt. Thank you.
And let's hear now from Ed Ryan.
Johannes, thank you for a wonderful, wonderful talk, and Mike, thank you very much for the invitation. So there's about 1,000 things, and maybe we can flow over into the reception. But I guess a couple of things. Paratyphi C is a strange bug, right? So there's all different salmonellas, and salmonellas. There's over 2,000 serovars, and they have, most of them are zoonotics. There's only actually 3 that are human restricted. That's Paratyphi A, Paratyphi B, and typhi itself. C is even an outlier in that it's a cause of typhoidal fever, in that the person can get a typhoidal illness, but it also can cause abscesses and sepsis, and it can persist in the body for a very long time period. There's multiple reports of people with Paratyphi C who have an abscess drained in their liver or their spleen, and then 30 years later, they have another abscess in their liver and spleen, and they've moved from wherever they came from, and they then have the exact same bug 30 years later. So it can persist the way that tuberculosis can, I guess, is one thing. Number 2, the patients go much more septoid, which means they get sicker. Most salmonellas we break into 2 categories. It either causes a gastroenteritis, or it goes deep. It has immune stealth mechanisms where it manipulates the immune system, and it actually doesn't cause a major diarrheal problem and goes deep in the body. And the question is, why did they do that evolutionary? Don't you want a lot of diarrhea so I can contaminate the water source or something? And we think what it is, and we're not sure, is that as we move out as migrations out of Africa, we actually moved in relatively smaller bands, and we didn't have the ability to keep infecting and keep the organism going. The typhoidal ones tend to hide long-term inside your body, inside of your gallbladder. And about 1 or 2% of people who survive Salmonella typhi, at least, is where our best data are from, and we think it's the equivalent with the others. 1 or 2% of those people get infected in the gallbladder, and they intermittently shed it through their biliary system back down through their stool, and that's the typhoidal Mary that Johannes mentioned. And we think that allowed it to then travel with our hunter-gatherer teams as they spread out and to introduce it into new environments, et cetera. And paratyphi C, though, doesn't really, it's a strange bug in that regard. You can get that chronic colonization and that chronic sort of shedding, but you have this more septoid features as well, which is a bit atypical. Now, we don't see, I guess what's strange here also is we don't see mortality to this level to paratyphi C. Where did this bug go? If, it was first reported in the modern era in a Serbian soldier in World War I. And it was reported, and then they started looking at these typhal malaria cases, and a subset of them, but not big outbreaks. The biggest outbreak that I'm aware of that was reported was in South Africa, that maybe had 100-ish or so over a 3-year time period in the Transvaal. And there was another outbreak in British Guiana back in the 1920s or so that had maybe 45 patients, et cetera. But even those large outbreaks were the logs different than what might have happened here. So, there's questions here of, and the other atypical thing about this is very closely related to cholera suis. And cholera suis, as Johannes mentioned, is a porcine pathogen. And De Soto introduced pigs to the New World in 1539 in Tampa. I don't know when it made it down to Oahu, but I guess the question is, what allowed this to circulate among the people there? Was it person to person? Would be a really high attack rate. Was it through the pigs? Were there pigs there at this time? I don't know if you can mention if there's any pig husbandry during this era. I don't know if you can talk about, could the pigs have, even with ancient DNA, could pigs from the 1540s have routed up the bodies and defecated into those areas? So yes, it might still be ancient DNA, but did they introduce it from their GI tract? Because cholera suis means the cholera of pigs. They don't get cholera in our sense, but it was a bad diarrheal illness of pigs, and they die. It's a veterinary problem, et cetera. So it's fascinating questions here. And I guess the question is, with your reads that identified this as paratyphi C, if this is paratyphi C that caused coccolizzi, either the bug has changed, or it was the people are different in that the group there had a very specific HLA type, et cetera, that they just didn't do well with the bug. And we can't really necessarily answer that latter one, but the first one is whatever read you hit for paratyphi C, it would be very interesting to see how they match up with modern day paratyphi Cs, because something is different about this bug, and 500 years ago is not that long ago. So it would be really interesting to see.
Yeah.
So lots of thoughts and lots of questions that we can have a chance to chat with at the break.
So, just to comment on the last part, we have actually looked at many more samples now, which are not part of this TPOS collection, but actually through the 3,000 genomes or genome-wide data sets that we have throughout human history, basically from all continents. And we have found it in Eurasia to be present over 8,000 years in quite a number of individuals. Places there are no pigs for sure, because they're from the steppe, from the Neolithic, that was even before domestication of pigs. And we also find, and that is very exciting, I think, for me at least, that if you look at the big diversity of Salmonella, of those 2,800 cellulars or genome-wide data is available, they all cluster with paratyphi C. We find nothing that falls anywhere else. They all cluster with paratyphi C. And that, I'm really puzzled by that, because it really seems to suggest that it has been quite prevalent in the past. We find it as much as we find Yersinia pestis, which we've also identified in many individuals, but they're all clustering together. So they seem to have something in common that causes some sort of enteric state that seems to kill people, because if we find it in larger amounts in those people, it could be that a pig just defecated on top of it, but they are also falling in different places, not just with cholera suis, but actually in different places in this whole branch of paratyphi C branch, basically. Also there's different close relatives that are present, which are actually today found in whales, for example, in turtles, in all kind of like chicken, for example, and other animals. So I don't really think that this is, I think there's a high chance finding it in such a high amount that it must have been present in that person. We also don't find DNA of other animals. We could actually, I'm not sure if we look for pig DNA in those samples. This is actually a good idea. We could.
Pig DNA
Yeah, I mean, we're comparing it to the whole gen bank now, so the pig genome would also be in there. So I think we would have seen it, but I can ask the people that did the work again if that actually happened. So I do think it is some sort of disease from the past, but it's a very intriguing and good question if it then changed somehow. And we have looked over this 8,000-year period now at virulence factors. And actually, the strains from TPOS as well as other strains going back to the Roman Empire, they have all the kind of virulence factors that we find in modern paratyphi C. So there's most of them are there. The cassettes are there, and it's quite complex. It's kind of different cassettes with basically pathogenicity islands. But then if you move further back in time, some are missing, some are gained. It's quite complicated because there's a lot of recombination actually happening through time. But yeah, we're currently kind of looking if there's something that they all have in common, which differentiates them from typhi and from basically all the other Salmonella, because they seem to have something that allows them maybe to become some sort of enteric state, for whatever that would be. And I'm quite interested to talk more about that if you have some insights on that, because it's really something we just discovered. This is unpublished, just a couple of weeks old, those results, and it's very, very exciting. We're just preparing a manuscript on that.
I was just curious how, if there were new virulence, I guess new isn't the right word, but virulence factors that are no longer found in the current strains, to what extent would you be able to find them given the hybrid capture, the hybrid selection approach? If there was just an entirely new cassette, I guess you might have found it from the original. Another way to ask this is how big are the fragments that you're pulling out? Might they have been attached to something you pulled down?
Yeah, it's an excellent question. If there could be things, for example, present that we don't have today in the genetic diversity, we would have a hard time to find them. And if they would all fall into this one branch and they would all be closely related to each other, I would kind of agree with you. The thing is that, and I should have brought a tree, I think I don't have one here. I could pull it up, but it would take me a few minutes. They're actually falling throughout the tree of the paratyphi, kind of like cerovars. So they're falling on many different branches. So then basically what would have had to happen is that this cassette or whatever was introduced in many different branches, but then lost in many different branches. And that's some sort of convergent loss and gain, and that's kind of unlikely to happen. So I don't think that this is a scenario that is possible. But of course, it could well be that we haven't understood all the pathogenicity and all the different virulence factors, and there could be some that we just don't know. And of course, there's recombination also, which makes that type of analysis also quite difficult, because you have lateral gene transfer between different cerovars, basically, that could have given some cassettes to all of them.
Sorry, a bit simple question, but I didn't hear, and I'm not a Salmonella medic. The drawings from that you showed,
yes,
versus the epidemiology of somebody who has this disease, let's say in Africa or Asia today, do they look the same?
So the question is basically, if you look at those codices and you see those kind of pictures of people kind of having some sort of bloody liquid coming out of their noses or faces, something like that. This is something that usually is not so common with that type of Salmonella, but there's certainly bloody diarrhea, which is quite common. Now the problem is, again, that's what Noreen mentioned, what I also said earlier on, that the historical accounts that we have are usually coming from the end of the 16th century, during the kind of the second Coalition, third Coalition. This is when basically people then wrote about what happened 30, 40 years, even 1 or 2 generations before. And it might just be that they used whatever happened in the late 16th century to infer what happened then, people getting sick. And there's also some cases in the literature you can actually find where people also have some sort of hemorrhagic fever-like symptoms, having this paratyphi C, and the kind of some septic stage where you also have some blood coming out of basically your mouth, and not just bloody diarrhea. So that can also happen in later stages of sepsis, of course. The body rash is a very typical symptom of Salmonella, so this is something that would fit quite well. So the only, I should have said that before, the only thing that doesn't really fit is the kind of basically blood coming out of your mouth. That would be a bit untypical, but all the other symptoms, the fever, the body rash, this is something that you find in a paratyphoid fever commonly.
Hi, thank you for your talk. I'm an archaeologist in New World contexts, and so I'm really excited to see these kinds of very innovative methodologies coming over here and doing kind of new things. I have a little bit of a different kind of question for you today. I read one of maybe the publication that came out of this work, and in listening to your talk today, I'm a little bit curious about the way that this work on disease is being contextualized within the broader literature on colonialism in the New World. For example, in your talk today, you refer to this sort of standard estimation of 95% depopulation that comes sort of from disease. In the paper, you note, whoever wrote the paper themselves notes that disease is an underappreciated factor in depopulation. In my opinion, I feel like it's somewhat overappreciated, and it allows us to sort of look past the violence that is done to these people. For example, in this context, it seems like potentially the answer to one of the questions of why is it so different here could be that, well, these people are being coerced into labor to build churches or something. So I guess I was just wondering if you could speak a little bit to how this work gets contextualized within the archaeological side of things.
Yeah, I mean, you're absolutely right, and it's often used as an excuse to say if 95% of the native population was killed by disease, then the poor people died because they got sick. So what can we do, right? But we don't talk about all the murder and all the mistreatment that those people actually suffered from. So I agree with you, I guess, in that regard. And I can just say that we're kind of relying on the historians and archaeologists that have been working on this topic in the past. And I think our research doesn't really contribute too much on kind of those estimates. If it's 95%, it's 50%, it's 20%, we can't really say that. We have a large cemetery with a lot of people that died likely of this disease that I talked about today. It doesn't really tell us about the numbers, and how you then kind of put that into the larger context of what happened in the 16th century, I think is probably kind of giving it back to you as archaeologists or historians to kind of use that type of data then maybe to see. I think as we discussed before, we need to explain why it's not so much of a kind of cause of death today, and it's not so much of a pathogen that's present today, but maybe in the past it had, as you said, higher mortality because of other reasons which come from the social context and not so much from the biology of the pathogen itself. And one could probably explore that even further. It could even be quite interesting to see under what circumstances can paratyphoid fever kind of be so high in its mortality. And maybe if we kind of look at historical accounts from the 19th or 20th century, we might actually find that there are cases in certain parts of the world where people were under certain conditions, imprisoned, for example, or outbreaks, or in other contexts that could then be applied to kind of the historical context of the 16th century in the New World. That might be a possibility, so it's a good idea.
If I may add my voice to that, your question is a very good one, and it underscores the importance of the point that Noritouros made about preserving the skeletons, having access to the skeletons, because one could imagine that in this particular case, it would be possible to examine the well-preserved skeletal remains, to look for signs of antemortem trauma, to see if this population was undergoing abuse, physical abuse, in order to look at their nutritional status to see whether they had the kind of very low nutritional status that's typical of so still having access to the individuals allows for the possibility of looking at this particular population for macroscopic or microscopic evidence of fragilize the population and could help to explain a very high mortality.
I have two questions. First, on the pathogen evolution, you've got, for some pathogens, you have some data extending over 5,000 years. What evolution did you see in those pathogens? Mm-hmm. And the second question is more a technical one. When you presented data from 1520 on of the population in Mexico, you had it decreasing from 23 million to 15 million in one epidemic. Where did those numbers come from? What's the evidence for them, and what are their uncertainties?
Yeah, I mean, there's high uncertainties, and as we just discussed, this is something that's been debated, and there's much much debate on that. That's basically coming from historical accounts. There wasn't a real census until the 1570s in Mexico, and then basically they tried to reconstruct what was there before, and then it comes from archaeology, comes from the few historical texts that we have from this time period, and then it could be seen as probably the highest estimate, the 95%, and some people might actually say it's more 50% or 10%. There's all kind of opinions. We do have historical accounts that of course cover the 16th century from Dominicans and other explorers, and of course those are kind of historical documents, so they should be treated carefully because they're subjective, they're not objective, so people might have actually used them in their favor. So this is, of course, a lot of uncertainty around those numbers. So it will be hard to come up with exact numbers. I think even kind of bioarchaeology will not be able to quantify exactly how many people it was. What we do have in terms of genetic kind of data is that people have actually looked at human DNA to see if we find any signal of this decrease in the 16th century. And we do find that, for example, in the mitochondrial DNA, there's quite a decrease of effective population size. So the genetic diversity is actually shrinking quite a bit. So we see a strong signal from the genetics that the population size have plummeted in the 16th century. But we can't really use that for a census. We can just see that there is a signal that the effective population size has decreased by a factor of 4. If that kind of can be directly calculated into census, I don't think so, but we do see a strong decrease also in the genetic data. Then your first question, if we look at long-term evolution of pathogens, so the one where we probably have the best understanding, which I mentioned before, is Yersinia pestis, which we kind of trace back now to about 5,500 years before present, where it's actually quite interesting, and we had a whole workshop dedicated to this morning here, where we talked about some of the research that we have been doing, some of the findings that we have, and it's quite fascinating. We have something like 100 or so genomes from the past, from 5,500 to basically the 18th century, and then compared to the modern genomes from all over the world that we have. And there's some really interesting patterns. So one kind of first thing that we notice is that the early form that we find in the late Neolithic, early Bronze Age, which is present, actually spreading with humans from the so-called Pontic steppe north of the Black Sea. It goes into the Altai as well, it goes into Europe. This form is lacking some of the virulence factors which are needed for the effective transmission through rodents and fleas. So it seems that this early form was actually not transmitted by fleas, which then suggests it was not bubonic plague, but it was some sort of other form. We have been debating it could be some gastric form that enters somehow into some sort of sepsis state, or it's some sort of pneumonic form that gets transmitted by droplet infection. We're not quite sure even how it got transmitted, but it was not in this bubonic stage. And then from about 3,800 years ago, we see the emergence of the bubonic form. We really see suddenly we have those virulence factors all in place, and this whole apparature for flea transmission via kind of rodent fleas into human population would be there present from 3,800 years, so from the Middle Bronze Age on. It's quite interesting, but then only in the Justinianic plague we actually pick it up in large-scale pandemics. So it's also something we debated this morning, why it took 2,000 years before it kind of causes major outbreaks within the human population. And then there's all kind of questions why it kind of disappears in Europe again and then comes back in the Black Death in the 14th century. But it's interesting to see those virulence factors emerging. But from 3,800 years ago, we have them all in place. So we see the early evolution from something that was not bubonic likely to something that was bubonic. So we really see evolution in situ, and actually the strains that were not bubonic, that kind of had not these genes, they're getting extinct. We lose them in the mid-Bronze Age when the other ones pop up. So there's some interesting kind of patterns there. So it's really fascinating to see this evolution happening in situ. And this is the one that we started about 8 years ago to do research on. Hopefully with kind of Salmonella and kind of other pathogens that we're studying, like leprosy, for example. We are now able to do similar things. We've done some things like that also for Treponema pallidum, so what causes syphilis, and Pallidum patinue that causes yaws, which we also now have from the past, actually also from Mexico from the 17th century. And there we also see there's actually antibiotic resistance popping up, but later in the 20th century, with strains that are then developing this antibiotic resistance, which are spreading. They're actually kind of emerging right now all over the world, which we also see happening. So this is also something that we can reconstruct with those ancient genomes. So, but I could probably go on with some other pathogens, but I don't have time for that.
Yes, thank you, Johannes. You mentioned two other pandemics in 1520 and 1576, and presumably there were a host of other epidemics of one kind or another. If and when we are able to get samples from the other pandemics and epidemics, do you expect Salmonella to be the pathogen in that case, in the same way that Yersinia pestis, I think we still understand, was the pathogen that returned regularly from 1348 onwards in Europe? Or would you expect perhaps to find different pathogens in those other cases? And in either case, why would you expect one or the other?
That is a very good question. What caused Kogelitsli 2, 3? Not having a strong expectation. I do think if you look at the kind of codices that were written later on, which show a lot of this kind of hemorrhagic type of fever that we see with the kind of blood coming out of the mouth, which we see in many depictions from that time period, I would not expect that to be Salmonella necessarily. So I do think that looks more like a hemorrhagic fever, which would be caused by some sort of filovirus or something like that. But then again, it could be just kind of a mispresentation and just kind of extreme cases, or people had a certain relationship to blood that they kind of depicted it in this way. But in that case, it would be an RNA virus, which would give us a hard time to identify it, because RNA viruses don't preserve, because RNA is not stable enough. So we would have a hard time to actually find that. In general, viruses are quite difficult because they have quite some similarity to mammalian genomes in many places, which mammalians actually incorporate a lot of viruses into their genome, so they are easily misidentified. But that will be very difficult to look at those RNA viruses. But we might find other things, smallpox, of course, which is a DNA virus, which we could potentially find. So far, we haven't been able to find. We have actually screened more material from that time period, from the late 16th century. We haven't found anything else, also not Salmonella. But I would hope that by screening more material, we one day will find also material from this time period. One should also say that there's not a whole lot of mass graves from the 16th century. So this goes a bit back to this question about, was it infectious diseases that diminished the human population, or whether it was maybe violence or something else. We don't have mass graves like we have during the Black Death in Europe all over the place. The typhus is actually the only mass grave we have from the Great Kogelitsli. And this was also one of the reviewers actually pointed out, what can we say from one mass grave about kind of larger pandemic? It's the only one. If we can't say anything from that one, then we can never say anything, because it's the only one that is actually known. I think also from kind of the late 16th century, we don't have a lot of mass graves. In general, in the whole New World, we don't really have mass graves like we have during the Black Death. So if 95% of the population has diminished, the question is, where are they, right? Where have they been buried? We probably have to find those bodies before we can look at those pathogens.
Well, I know that there's a lot more questions out there, but I would propose, rather than finding bodies, we go find some refreshments and continue the conversation in the room next door. But before that, let's thank Professor Krause, Professor Turos, and